All right, it's been just a couple minutes for me. I'm not sure if it's been days or so for you, but we're going to pick up where we left off with chapter 11. We really have to talk about how all these phospholipids fit together into membranes. Remember, they have two legs and a head group. And so what you're going to do is you're going to line up all of those like they're little toy soldiers, like I said, in the army. It reminds me of, I keep saying that because it reminds me of like all the stormtroopers in like The Last Jedi and things like that when they're all lined up on the deck of the Star Destroyer. Again, to each his own. The thing about phospholipids is that they tend to form straight bilayers rather than curved micelles. If you look at the difference here, if you have one tail, one leg to your fatty acid or whatever, you have a large head group that tends to be wider than the rest of the molecule. You have a wedge. When you line up a bunch of wedges, you end up with a circular micelle with like a spherical pattern to it. When you have phospholipids, you have two legs, and so you have the width of the head group is about the same as the width of the rest of the molecule. You have something that's like a cylinder, and then when you pack it up, you end up with it forming a flat molecule that needs to have two layers to be able to hide all the hydrophobics. So you need to have an outside and an inside layer that come together with hydrophobic in the middle. You're used to how this looks when you look at like when we've shown membranes before, and they look like this. And it gives them the three-layer sort of appearance because you have a hydrophilic layer, then a hydrophobic layer, and a hydrophilic layer on the bottom. The story that I always tell about Aiden at this time is that, so Aiden is my second boy, he's the redhead, and he actually had a large head growing up. And he was also tall and skinny. And so he really looked more like a wedge-shaped micelle, and he took a long time learning to walk because he was so top-heavy, and it's literally true. He was so top-heavy that he would fall over when he was trying to walk. And so it was like a year and a half before he figured out how to put it all together and how to walk without falling over. Now that he is more normally proportioned, he has grown up and his body has gotten wider and his head no longer makes him fall over. So now if we took a bunch of Aidens, used to be if we took a bunch of baby Aidens, we'd make a micelle. If we take a bunch of teenage Aidens, they'll make a nice little bilayer, and that bilayer would be colored red because, like I said, he's a redhead. So you can think about him as having a red-colored head group. I'm not going to be able to tell that story because I'm sure it will embarrass him terribly in the future. But it was so cute, you know, anyways. And he's a great kid. So if you have enough of the bilayer, it can eventually curve around on itself. It just makes a larger curved structure, and it makes a curved structure with an inside that is hydrophilic. And this is what a cell membrane really is. A small version of this is called a liposome, and a large version of this is called a cell membrane. Depends on the size of the cavity that you have. So when you have membranes, you have this three-layer appearance if you stain them with something hydrophilic. Osmium is a charged metal. It's very heavy, so it shows up in electron microscopy very easily. If you put osmium on a cell and look at the cross-section, you see a three-layer sort of sandwich sort of appearance, where you have a dark hydrophilic layer on the outside, a light hydrophobic area where you have no osmium staining in the middle, and on the inside you can see there's a slightly darker hydrophilic layer again. These are the head groups. So the osmium associates with the head groups, none of it gets into the middle. This shows you how the membrane bilayer acts as a really nice wall. So phospholipids, proteins, and sterols, and glycolipids, they all add together and they form membranes. Okay, so you have you have a lot of things in the membrane. Like we said, cholesterol can actually go in the membrane. If you look down there, it says sterol. Because remember, cholesterol actually has the same shape. It fits into the membrane. You have a bunch of other things as well. You can have the phospholipids with glycolipid groups on them. The glycolipid head group will be pointing up, and so you see that's on the outside of the membrane. So you have this bilayer organizes the entire thing. The important thing to remember about this bilayer, this is just a picture of it. There's nothing holding each of those little toy soldiers in place, each of those phospholipids in place. except for non-specific hydrophobic interactions. So it's like there's a two-dimensional liquid of these flowing around each other. They're marching around like a crowd of people. And so you have a lot of fluidity to the membrane. It's just fluidity that's held in two dimensions by the fact that you have the bilayer structure holding everything together. You can also have different types of lipids, and sometimes those different types of lipids will mix together cleanly, and sometimes those different types of lipids will separate out. And so you see right here, you have a bunch of green sphingolipids on the inside layer, and they're all gotten together because their hydrophobic, hydrophilic interactions work out so that they tend to associate more with each other than with the other lipids in the membrane. So what you have is you have what's called a lipid raft, because it's kind of like they all sit together and they form this sphingolipid area that is just a little chunk of the membrane that is all sphingolipids. It's just a matter of what dissolves with what. And you have complex interactions, we can observe how those work out. So when you have a complex membrane, you can put stuff in it. And in fact, you can make a big cell membrane out of phospholipids. You can also make a little liposome-sized thing. And it turns out that liposome-sized little bubbles of membrane lipids are actually used to send little escape pods of material around the body. These are called exosomes, and this is a really good example of what one looks like. Because the body sometimes uses these to send messages and material from one cell to another, drug designers have started to look at it as well. Pharmaceutical companies have started to say, well, can't we use the same thing? It turns out that the right kind of lipids will self-organize into little liposomes like this, and you can put something inside it. If you look carefully, you can see there's a lot of nucleic acids inside this exosome. You can put this in a body, you can inject this into a patient, and it can actually flow through the bloodstream. And you can even put, like, you can see there's a couple of membrane proteins or something in there. You can put particular targeting proteins in there so they go to a certain organ. This is actually really cool, but again, it works on the way that phospholipids work. Here's a side view of many different of the ways that a membrane can come together. And just remember, if you have, depending on the saturation of the membrane lipids themselves, you'll have more liquid or more solid characteristics to the whole membrane. If you have completely saturated chains that are all in the membrane, you're going to have a very non-fluid membrane. It's going to be a lot closer to being solid for the same reason that saturated beef fat is solid. It'll be like a beef fat membrane. If you have more unsaturated points, it's going to be an olive oil type of membrane. So it's going to be very fluid. And if you look over here on the right, if you mix sterols in, the sterols can fit together, but they are more like unsaturated than saturated chains. And so they're going to end up making the membrane more liquid. And so that all works together to keep the membranes liquid. It turns out a liquid membrane is better for life. Cholesterol allowed this to happen. So this is a slide showing that you can have a membrane that can curve. Not all cells are nice little spheres. When you have this fluid membrane, they can actually like make little pseudopods, and the way that amoebas move around and they reach out for stuff, well, that's really changing the curvature of the membrane. And it's taking that liquid, oily membrane and reshaping it by some proteins inside the cell or whatever. The important thing is that you can't just do this with regular old phospholipids. You need the right liquidity of the membrane, and the molecule that really gives you the right mixture of flexibility and strength is cholesterol. Turns out that cholesterol in the membrane makes it just liquid enough and just strong enough to be able to make weird sort of shapes like this. Those shapes are important to life, and that is why cholesterol is a good thing for life. It looks like when cholesterol was evolved, which, by the way, required oxygen to make cholesterol, because remember you had that little OH group on the end. So after oxygen increased, cholesterol evolved. Cholesterol allowed life to do new things. And that's one of the arguments that I make for why cholesterol is a good thing and oxygen is a good thing, and the two of them go together. If you think about this, they don't have to be like the little exosomes that you send out, don't have to be just little spheres like what they showed in the thing, like little spheres like this. But you can put a bunch of the spheres together, And you can join them together, and then they'll make a little cell membrane of their own. And so there's ways in which cells have done this. There's a bacterium in a biofilm that is talking to its neighbors by putting a bunch of exosomes together and making a long chain that's kind of like an airlock or like a bacterial lock for passing stuff from the inside of one membrane to the other. They're literally building bridges to each other. It's one of the reasons why biofilms are so nasty and so hard to destroy. The bacteria are using these flexible membranes to form these interconnected things. Is this one bacterium, or is it acting more like a multi-celled organism? I think it looks more like a multi-celled organism, but it's somewhere in between the two. If you look at the formation of these, you can see the bubbles in part D that end up forming a chain in part E and F, and the chain can connect to bacteria. It's all membranes flowing into each other and dissolving in each other. So tumors can even use this, not important just for bacteria, but actually tumors form these microtubes to each other, and they pass calcium around, and by passing calcium around, they survive chemo better because the calcium helps them survive the rigors of chemo, the cell death that chemo causes. If we can block these microtubes, we block the calcium, and we help facilitate the chemo because we help the chemo kill the tumor cell. So I want you to have this picture in your head. I want you to have the picture of how a membrane moves. And here is an actual picture of a membrane moving right here. It won 3rd place in the 2011 Bioscapes contest, so I want you to look at this. And that is the membrane moving. You can see that the membrane is flowing like a liquid, but it's clearly a hollow sphere, flattened out in the case of this cell, but it's definitely flowing. And I want you to have that image because the membrane is only held together by non-specific hydrophobic interactions. Liquid interactions hold the membrane together. And so the main thing I want you to see, membranes flow like oceans. If you look at this, this is a way that they've sort of traced how the membranes flow in a cell. And they even have like little whirlpools like oceans do. This is a very liquid case. I'm going to go ahead and stop this because I'm going to make sure that this is working before I get too far out. And so I will put up 2 lectures instead of 1. Double your buy one, get one free.